Hello everyone. Today I'll be presenting you all a research topic based on genomic analysis of group B streptococcus. The studies were conducted on camels from Africa. As we all know that Africa is a very deserted area and 50% of the diet of the people living there it comes from the camel milk and meat. Camels are very well adapted to the harsh conditions and they can produce milk in very large quantities despite of having the lowest nutrients present in the body. The pastoralists that raise camels over there, they consume the milk that is unpasteurized and that poses a risk for zoonotic disease. Kenya is the third largest country in camel population in the world, and the most common disease associated with this camel is mastitis inflammation. It is the inflammation of mammary glands of the camel and is caused due to the group B streptococcus bacteria. So we'll be discussing about this. This whole research is based on this bacteria and how it is isolated from the mammary glands of the camel. Methods like how we did this research, I'll just explain you all. There are various steps to investigate the molecular epidemiology of GBS in dairy camel herds in Kenya using genomic phylogenetic analysis or spatial mapping. But the steps used here are in this particular research, the steps that were used were, first of all, the study area, that is the mammary glands of the camel, and the selection of particular herds of the camel. It was focused. Then how the milk was sampled from that particular herd. After the sampling of the milk, the bacteriological culture was isolated, and then the DNA extraction was took. DNA was extracted and sequenced. This was known as a phylogenetic and statistical analysis. So all of this together gave us the few results, and that will be discussed later on. Focusing upon study area and selection of herds, I would say that the study was conducted in a particular town of Kenya, central Kenya, that receives an annual rainfall of approximately 150 to 250 millimeters. These camels, they generate the milk for the consumption of the whole town. But the milk market is growing over there, and officially the milk chain is also growing. But the herds that were selected, they were at the end of the dry season or the early wet season, not during the rainy season. The data was collected and it was stored digitally. As per the data, the camel milk sold with end markets in that town and central Nairobi town. So let's see how the selection of herds helped us in going through the further analysis. Now, after the selection of herd, we sampled the milk, the bacteriological culture, and the isolated selection. 7 to 13 camels per herd were scanned from 20 pastoralists, and their milk samples were collected to check the mastitis inflammations among them. First of all, they were initially checked for non-milk producing quarters of the glands and the signs of inflammation that were shown in that particular gland. After that, the milk from that particular quarter, it was sent to the CMT, to the processing. CMT means California Mastitis Test. It's a particular test. It's an evaluation of basically milk leukocyte count. So it can just let us know that how much severe the inflammation is in that particular area of the gland. Milk, first of all, the milk was it was just visually inspected. Like we can just go for the color, the viscosity, blood, if there is any blood present in the milk or some clots are present. So first we just had an basic evaluation of the milk sample, and then we sent the milk sample for the CMT test, for California mastitis test. After that, what was done? The liquids, they were gently mixed with the paddle. The viscosity was measured from scale 1 to 5. After that, this viscosity measurement, primary species identification, it was like 24 to 48 hours incubation time was given to for the bacteria to be grown, incubated, and final detection was confirmed of the species. Mass spectrometry was finally used to detect the species. Now, how can we say that first of all, this is the milk sample. There can be two cases. If the CMT is less than 3, the CMT value, the mastitis test value is less than 3, then no mastitis is there. We won't consider that. If the test value is greater than or equal to 3, then we'll consider it. So for the milk sample that was collected, out of the 12, I would say that 12 camels, they had no mastitis, and the other 53 had mastitis. From those 53, mastitis was two types, like subclinical and clinical. So subclinical mastitis, there were 40 camels, and other rest 13 camels, they went for clinical mastitis. Clinical mastitis was further divided into two types, acute and chronic. Acute one is like, that is, everybody knows what acute means, like for a shorter duration of time. So acute clinical mastitis, just for 8 camels, and the chronic was 5 camels in that category. So here I can just show you, like from this flow chart, how the milk sample was further classified for the disease. After that, all of these studies, we went for DNA extraction and sequencing. The GBS colony material, it was collected with the calibrated loop of 1 microliters and suspended in a 600 microliter nuclease-free water for DNA extraction, just like we normally do in the labs. This suspension was mixed with 0.1 millimeter of silica beads, and it was added to the fast prep 24, and it was run at 6.5 meters per second for 3 2-minute cycles were given for this thing, like total 6 minutes. Then from this 200 microliters, DNA was extracted. DNA concentration was measured using a fluorometer, and it was adjusted to like 7.5 nanograms per microliter. Further, like I would say, this antimicrobial susceptibility and virulence testing was conducted for this. Okay, a PCR targeting like lactooperon, everything, for the presence or absence. After that, phylogenetic and statistical analysis. What comes in this? All the assembled genomes, like they went through a new alleles and submitted like for allele number and ST assignment. A core genome alignment was obtained. A map of herd coordinates, like you can see this flow chart basically. All these results, based on this method to find the presence of GBS, following results were concluded. The sequence types and serotypes associated with camel mastitis, antimicrobial susceptibility and virulence testing, phylogenetic analysis. These were the 3 results associated with all of these studies from camels. Sequence types and serotypes associated with camel mastitis, they were like quite in scientific detail. So basically, yeah, so I'll go for antimicrobial susceptibility and virulence testing, like how phenotypic susceptibility testing revealed tetracycline resistance. The TET-M gene was found in all phenotypically tetracycline resistant isolates and was absent from the 2 isolates that were phenotypically susceptible to tetracycline. Apart from the TET-M gene, no more genes coding for antibiotic resistance were found within the GBS genomes. And like after that, all the PCR results were 100% in agreement with the findings of phenotypic lactose fermentation and the presence of a lactose operon in the genomic analysis. For mastitis isolates, that is the ones that were associated with the inflammatory response, LAC2D was overrepresented. The human associated virulence genes SCPB and LMB were detected in a single genome assembly which belonged to ST1 as a part of known composite transposon. And what comes in phylogenetic analysis? After all this initial inspection, the isolate belonging to ST1 was removed from the phylogenetic tree to better visualize the relatedness among the other isolates. 3 main lineages were observed in the core genome phylogenetic tree. 2 corresponded to a single ST, whereas the third and the largest lineage included ST616 and its SLVs ST1653 and 1654. Within the major lineage, pairwise genetic distances such as the number of single nucleotide polymorphisms and between isolates did not differ within the herd versus between herd comparisons. So like there was not much difference. Final discussion, it focuses like how the based on the results there were three results basically. Like, how can I discuss? The genomic analysis, it showed the existence of a predominant niche adapted strain in camel milk. Then secondly, camel GBS isolates from different herds are genetically related, because it's basically coming from a single species of camels. That's why it's similar. Then I'll go for potential interspecies transmission. Genomic analysis showed the existence of a predominant niche adapted strain. How it, like, it's same. It's here. After that, camel isolates from different herds are genetically. I can explain you like how it is. Um, and after that, like, we'll go for potential interspecies transmission. Like, it's how the bacteria transmits between the species. Finally, it can be concluded that the GBS discovered corresponded to a single sequence type ST616 or its single locus variations within a substantial portion that displayed evidence of niche adaptation from the samples that were collected from the Iolo town of the Kenya country. As a result, the mastitis causing GBS strains in camels, they are expected to be primarily udder bound, highlighting the need for enhanced milking hygiene that needs to be practiced by the pastoralists. Because the heterogeneity of isolates inside and between the herds is similar, internal and external biosecurity measures are essential to limit the transmission within and between the herds respectively. And finally, I would like to add here, that the discovery of one presumably human-derived isolate ST1 in an infected udder clearly suggests that human to camel transmission is a possibility, and this danger should be investigated further for the strains to be present, for the bacterial strains present. And here I'll end my presentation, and thank you so much for listening to this. Thank you.